Okay. So I got the recording going. Welcome, guys. I know it's been a weird schedule, so I just want to remind everyone about the schedule for the next few days. So today, again, obviously in-person lab is canceled. We're having severe weather this afternoon, and so I wanted to make sure both the morning session and the afternoon session, we were doing kind of the same thing. So because of that, we're not going to be able to work with the blackworms today. And so I switched the order around, and we're going to talk about how to use Excel this morning. And it's not ideal because ideally you would have generated some data in 2 weeks worth of labs to then practice with using Excel. But that's not the case. So we're just going to learn Excel, and then we'll go right into the Blackworm labs starting next week. As a reminder, tomorrow is not journal club because Wednesday was a wellness day. The students who normally meet Wednesday, we're going to meet virtually tomorrow on Zoom, and I will send out that Zoom link when I finish the recording here and have all of that completed. I also may have to jump off briefly and pause this briefly. I have a student from another class who has to take a makeup exam this morning. And so at some point I will set her up with her exam, and then I'll come back and we'll keep talking about Excel. Okay, so what I wanted to do is I'm going to share Canvas, and we're just going to talk through a few things first, and then we'll get to it. If you have questions, please put them in the chat or unmute yourself and ask. Okay. So we finished module 1. So some things coming up. These are reminders. These are the reminders I sent you guys. Hopefully you were able to fill out that chart that was due 9 a.m. this morning. Basically, it asked you to read through the lab handout, watch a short video, and then fill out a chart about what effect different chemicals are going to have on heart rate. The lab attendance quiz, I'll get to that in a second. I'm going to pretty much give everyone points for that. And then class 6 attendance, that will be for tomorrow's group in the virtual class. You've got a reading assignment. You also have a reading assignment that's about statistics that will help make today's class make more sense. So please do these 2 reading assignments this weekend, and then your pacemaker cell homework is also due on Sunday. Okay, so we're here, we're in our module 2. I had one last student take a makeup exam this morning, so my hope is to get your exam grades on Canvas sometime tomorrow. So that will be done. Okay, so if you scroll down, and it's our lab, it's technically lab 6, but we're doing it as lab 4 because that's just the way we roll. Let me see if I can move this bar. Hold on one second, guys. Move this bar down. Okay. So if you're in your lab 6 agenda, again, this is the Zoom link that we've been using. If you want to click on the lab 6 attendance quiz, all I'm going to ask everyone, so if you're watching the video at a later time, or if you're currently here, if you want to click on the lab 6 attendance quiz and then just write the time that you're currently watching this. So for students right now, you can write 9:32 or 9:33 a.m. Okay. And if you're watching this recording later, just write the time, just so I have a rough idea of who watched the video when. Okay. And then what we're going to be doing is we're going to be talking about Excel using these 3 tutorials. Now, these are the same tutorials. If you've taken Bio 4700, if you took it with me, I use these tutorials. I got them from Dr. Rock. So if you took it with her, I believe she uses the exact same tutorials. And I believe Dr. White also uses a similar tutorial. So you may have seen this material before, okay? That's okay. I think it's important to go through it one more time. So I'm going to ask you guys to download these 3 documents, download them to your computer, okay? So while you take your attendance quiz, just tell me the time and then download these documents. I want to remind you guys about a few long-term things that are coming up. Okay, and for that, we're going to scroll down. So I put together the information for your research paper, okay, as a reminder. Let's just look at this briefly. Your references are due on the 28th of March. You're writing a research paper about a disease related to the gut microbiome that interests you using scientific journal articles, primary research articles. So on the 28th, all I would like you to do is to submit 3 research articles, okay? What that is going to do for me is I'm going to check those and make sure, 1, they're all related to each other, and 2, that they're actually a primary research article and not a review article or a meta-analysis. If you are confused about what a primary research article is, please email me and I can clarify that a little bit. So due on the 28th are your references for your final paper. Your draft is going to be due on the 11th of April, so in less than a month. And then I'm going to give you comments on that draft. You're going to revise it, and you're going to bring it to class on either May 3rd or May 5th, and we're going to have a writing workshop where you will work with someone in class to edit your draft, and the final paper will be due during final period. Okay, so this is something that is a longer term project that has a due date coming up, so I want to put that onto your radar. The other thing I want to put onto your radar is this, the summary and analysis one is also due March 28th. So I'm asking that you go through, if you click on this, you go through and you write a summary and analysis between 800 to 1200 words using this guide and double checking it with this rubric. And it has to be about someone else's paper that they've presented. Okay, so not your personal paper that you worked on for Journal Club if you've already presented, but someone else's. Okay, so that is what I would like you to do. You have to do your first one by March 28th and your last one by the end of the year. Okay, so those are some long-term due dates I wanted to throw onto your screen here. Give me 1 second. Okay, wonderful. Okay, those are any questions about those long-term due dates, the schedule, life? Okay, attendance quiz question again is the time, the current time. What we're going to do now is I want to share my screen and we're going to walk through some of these tutorials. And so the best way to do this, let me pull up my chat so I have it. The best way to do this, guys, is to have this page, let me minimize it so it doesn't take up the whole screen here, is to have this page open and to, hold on, give me 1 second, let me move this here. I'm moving some stuff around. The best way to do this is to have open, give me 1 second, guys, I apologize, to have this open on your computer and to do it along with me, okay? To do it along with me. So I'm going to make sure I have all of my windows open because that is important. And I'm going to move my stuff around here. Okay, awesome. So we're going to start with the first one. And this tutorial is going to help you just get familiar with Excel because I know there are some people who are not familiar with Excel. And my goal is for this session to not take more than an hour, mainly because I want to make sure that I personally get home on time and that I'm not here super late. So let's get started. So if you've ever has opened up the tutorial 1 basics, we're going to go through. So if you click on the first tab here on the bottom, we're going to enter numbers. Oh, first, let's do on the intro. This is going to tell you what everything is. Okay. You're supposed to technically maximize it to full screen. I'm not doing that because I want to have my chat open so I can see people's questions. Okay. We're going to put numbers into a box. So throughout this tutorial, you're going to be asked to do questions, and they're going to appear. Anytime you put answer, they will appear on the answer page at the end. They're going to populate the answer page. So what I would like for you to write is your name in the green box where it says a name.
Dr. Alexander.
Yes.
Do you think you can give us permission to record?
Yes. How do I do that?
I don't know. It just said record on this computer, and I was like trying to, and it said, please ask the host to give permission to record. Okay.
I don't know how to do that. I'm recording, and I'm going to post this video as soon as it downloads.
Okay. Thank you. I understand.
Yeah.
That's fine.
Okay. I wish I knew how to do what you asked, but I don't. Okay. So the first thing we're going to do is we're going to take these numbers, and I would like for you just to enter them into the box, into the blue box. So we're going to enter them in a running tab. You're going to hit enter at the end of each one of them. And in Excel, if it detects a number, it's going to align it to the right. And so you know that you've entered the number correctly if it shifts to the right. Now, if I instead accidentally did a space instead of a period and I hit enter, you see how it didn't shift it to the right? So that's saying this is not a number. Okay, so we're just going to do this, enter these numbers in here. Bam, 18.6, 32.7. Okay, this is the basics of Excel. So now we're going to click on the next tab at the bottom where it says highlight. Okay, we want to highlight the select field. So in order to highlight the first cell, we're going to click on it, and then you're going to kind of hold and drag. Okay, and this will allow you to highlight the whole selection. So you're going to highlight all the numbers in blue. Okay, and then we're going to copy them and paste them into the yellow field. So you're going to hit Control C, or you can do Edit Copy if you would like. And then I'm going to click on the yellow box and I'm going to hit Control V. And look at that. I have now pasted those numbers. Okay, but sometimes we want to do this without the middle column. Like we want to just do the two outer columns. So if you scroll down, what we're going to do is we're going to highlight the first column. And then before we let go of our mouse, we're going to hold the Control button. And then we're going to highlight the second column. Okay, so again, click and drag. And before you let go of the mouse, hit the Control. And while holding the Control, click and drag for the next one. And then we're going to do the same thing. Control C for copy. Click on where we want it to go. Control V for paste. And look at that. The pink column has gone away, right? We have just copied these outer two columns. Okay. So what I would like for you to do in the answer key is to put what happened. Were they separated by a column, or were they now side by side? What happened when you personally did this endeavor? Okay, great. So we're going to do means now. Now that we have the basics of maneuvering in Excel, let's see if we can do some of the traditional math of Excel. This is what Excel is really good at, doing math for you. So the first thing we're going to calculate is a mean, which is an average, right? So anytime you want to do a calculation, you need to start with an equal sign. So we're going to hit equals, and then we're going to write the word average. And as soon as you start typing, Excel is going to try and predict what you mean, right? So you can either keep writing the word average, or you can double click on in the menu what you mean. Okay. And now for the average, we want to do all of these numbers. We're going to do the same highlighting. We're going to click on the first one and drag and highlight all of them. Or we could type it in, D5 colon D11. And the colon means through. So starting in the cell number D5 through the cell number D11. And importantly, we have to close the parentheses. We have to close the parentheses, and then we're going to hit enter. And it has now calculated the mean of these numbers. How cool is that? If you did it correctly, you should get 27.2. Take a minute and see if you can remember, what does the mean tell us? What does the mean? What do I mean when I say mean? What does the mean mean? Okay, take a minute. Any questions up until now? You can chat or you can unmute yourself and ask.
Yes, you said we're doing the first one, right? The Excel tutorial.
Excel tutorial 1.
Where I download the file and I clicked on it and I opened Excel. It's giving me something else. I don't know what it is. Like it's not the same thing you use.
So does the first page look like this?
Yes, on Canvas. As soon as I downloaded my laptop and open it up, and it's just something else.
You're not getting this?
Yeah,
No.
That's the same thing for me.
You're not getting anything close to this?
Like it's
No,
like
not at all.
a kind of like a blank screen.
Oh man. Okay, let me stop sharing for a second. Let's see if we can troubleshoot this. Alyssa or Kevin, can one of you share your screen so I can see what the heck we're looking at?
Yes.
Okay.
Whoever gets to it first gets the privilege. For other people, are you able to follow along? Is it just Alyssa and Kevin who are having this issue?
So can you see that?
Yeah, okay. You're on the wrong tab. Alyssa, on the bottom left, it says intro. On the bottom left, you see intro? Click on that.
Oh, okay.
Now, we've been moving through the tabs. So right now we're on the tab that says means.
Okay, sorry.
It's okay. Kevin, were you having the same problem?
Yeah.
Yeah, okay. Let's you stop sharing and we'll take back over. Okay. That's okay, guys. So we're moving through the tabs on the bottom. Okay, I'm gonna share my screen again. I'm taking back over. Okay, so on the bottom, wait 1 second. You were sharing your screen. Let me pull the chat up. Wait 1 second, guys. Is anyone asked something on the chat? I want to have that. Okay, so on the bottom here, I've clicked intro, we've done enter. We've done highlight, we've done means. We're now going to do standard deviation. Okay, so to do standard deviation, we also need to do a formula. So a formula, we're always going to start with an equals, and then we're going to start typing ST, and again, a whole bunch of stuff is going to come out. What we want is the standard DEVA. So you're going to double click on that one, and you're going to do the same thing. You're going to hold down and highlight the column, and you're going to close the parenthesis, and then you're going to hit enter. And you should get around 6.3. Okay. So we've calculated the mean, we've calculated the standard deviation. These are some of the most used statistics in biology. And so these are really going to be like your bread and butter formulas for using Excel. Okay, let's do a few more tricks to make things easier. Let's fill. So in Excel, you don't have to type things over and over again. We're going to type them once. So we're going to hit equals. The average, we start typing, we see average, we double click. We click and drag the numbers we want to find the average of. We close the parentheses, we hit enter. Now we're going to do the standard deviation. Standard dev, I see the one I want. I double click. I click and hold and drag and see that whole column. And then I close the parentheses and I hit enter. Okay. Now, if we want to do the same thing in every single column, right? So what we did in the first column, we want to do on the second column, the third column, and the fourth column, we're going to do a shortcut. So we're going to click and hold and highlight the mean and the standard deviation. And if you go to the bottom right corner, your cursor should change from a white cross to a skinny black cross. When you get that skinny black cross, you're gonna click and hold, and then drag all the way across. And what you've done is you've populated these cells with the formula to the left. And you can check this by clicking on the box and looking on the top, and it will tell you what is happening. So this is doing the average of E4 to E10. E4 to E10. Awesome. That's exactly what I want. So I'm going to close that. This is doing the standard deviation of E4 to E10, and so on and so forth. So rather than typing, fill lets you populate multiple boxes with the same formula. Okay. So in addition to fill right, you can also fill down. So I want you to take a minute and see if you can think of a reason when you would ever fill down, when you would ever do that, but dragging it down. Any questions at this point? Okay. If you have questions, that's what I'm here for. This is the time. Okay, so let's do pasting. Let's have fun with pasting. So we're clicking on the next tab here. The joys of copy and pasting. So if you have two similar charts and you want to perform the same calculations, so I want to find the mean and standard deviation for all of these columns, and I also want to do it for these columns. Copy and paste will do that. Copy and paste for Excel is defaulted to formulas. Copy and paste on Excel is defaulted to formulas. It assumes you want to copy the formula more than you want to copy the exact number. I'll show you what I mean. So I'm going to do what we just did, equals average. I start typing, I see what I want, I double click, I click and I highlight, I close the parentheses, I hit enter. Equals standard dev. I see the one I want, diva. I double click, I click and highlight, I close the parentheses, I hit enter. Now I want to fill them to the right to do both the mean and the standard deviation for all of these columns of numbers. So I'm going to highlight my formula cells. I'm going to get that black skinny cross on the right lower corner. I'm going to click and drag it to fill to the right. Now, while everything is highlighted, I'm going to hit Control C for copy. And if I click down here on that mean box, the first one, I'm gonna hit Control V for paste. Oh my gosh, look at what just happened. To find out what's happening in that cell, you look up here on the formula bar. It is doing exactly what I want, the average from D14 to D20. So it copied and pasted the formula and applied it to this new set of data. Okay, so everyone, please do that step. Any questions about pasting? Okay, we're moving on. This is a super fun, I know this is a super fun class, but hopefully this is useful. If you click on PS1, we're gonna do some special pastes, So say you put your data accidentally in a horizontal way, in a row, but really you want it to go vertically in a column. We can fix that by doing a special paste. So I'm gonna highlight the data. I'm gonna hit Control C. I'm gonna then click on the box where I want the top left of that data to appear, and I'm gonna right click. Oh, look at all these fun options that just appeared for paste special. I want a special paste. Now what I'm looking for is paste transpose. And so paste no borders, paste transpose. There it is. I'm gonna click on that. Look at this, look at this. I've taken the data from a horizontal row to a vertical column. Bam, look at how fun that is. So paste transpose, again, hitting paste special and then clicking transpose will flip your data. It'll rotate it 90 degrees. And so it will take horizontal data and make it vertical. Okay, how exciting is that? Any questions about paste special transpose? Okay, we're moving on. We're making great progress, guys. Let's find another paste special. Okay, let's say I did my normal calculations here, mean, standard dev, but I want to just paste these numbers. If I highlight them and hit Control C, and I go to the yellow box where I want to put those numbers, and I hit Control V, oh my gosh, something terrible has happened. Let's see if we can figure out why. If I click on this and it tells me, oh, it's trying to take the average from C9 to C15. It thinks I have another column of numbers because again, in Excel, it pastes the formula. It doesn't paste the number as the default. And there are no numbers here. And so it's like, what are you trying to do, woman? This is messed up. Okay, so instead we have to do a paste special. So we're gonna do it here in this pink box. We're gonna right click, we're gonna hit paste special. And we want to hit the one that is just the values, just the numbers. Bam. So what it has done is it has copied not the formula, but the number. So what you see here, bam, now the numbers. Okay. So paste special, if you paste special values, now you've just pasted the numbers. Okay. So in order to analyze data that you collect from an experiment, you're going to need to set up a data table. And this is what you're going to need to do for your Blackworm data. You have to set up data tables. So generally, you want to put them into columns. You want to have data in columns. And you want to break and separate your control group from your experimental group. So this is, we're imagining I have one control and then one experimental group down here. And I've measured 3 different things. Okay, let's say number of adults, length of latent phase, growth, whatever. I've measured 3 different things and I've measured them 5 times. So these are all repeats, right? I've measured one thing, 1, 2, 3, 4, 5, 1, 2, 3, 4, 5, 1, 2, 3, 4, 5, 1, 2, 3, 4, 5. And I've set it up so that when I put the numbers in, bam, I'm gonna get the mean and the standard deviation. And I've done that just the same way with the fill and then the copy paste. Okay, so this is kind of how you'll need to set up your data table for your Blackworm data. We're going to be measuring things. You want to have a control group and you'll have an experimental group and you'll take repeat variables. And you want to keep the repeats in columns because it makes the statistics look nice. Okay, so if you've been answering things in the green, the last page is the answers page. And so what this will do is if you put them in the green, it will put numbers here or words where each of your things are. And you can see your name has also populated. Okay. So if I was doing this for credit, I would screenshot this page. I'm not going to be grading these because of all of the weather stuff that's happening and because we don't have our own data to play with. So this is just for your own edification. Okay, that was Excel tutorial 1. We're going to move on to Excel tutorial 2. I'm not going to save this. Are there any questions about Excel tutorial 1? Going once, going twice. Okay, we're moving on. We're going to try Excel tutorial 2. Okay, so again, give me 1 second. Let me minimize. Oh, minimize this. Get it to the right size. Give me 1 second, guys. 1 second. Adjust, adjust, adjust. I'm going to open up the chat. Okay. It's the same thing on the bottom. So if you opened up on the very end, you're going to need to move. Like say when you downloaded it, you ended up on this page, right? And you're like, what do I do? Okay, it's okay. On the bottom left, you should see this little arrow. Click this arrow. Keep clicking, keep clicking, keep clicking. And you should get to the intro. I did, I got to the intro. Great. Okay. We're going to be doing some math. Now, we're not going to do all of this. We're just going to do some of it. Put your name where it says name on the name page. Now, we're going to have to install a program. If you are using a Mac, this is not going to work. So if you're using a Mac, you're about to reach a series of errors. And the newest version of the software for Macs, there is no solution to these problems. So that's why for this, I'm going to go quickly because I assume a lot of people are on a Mac. If you actually just want to put in the chat what kind of device you're doing this on, if you want to put in the chat what kind of device you're doing this on, if you're doing this in real time. For those of you watching the video afterwards, if you're using a Mac, you're just going to skip ahead. Let's see what we got here. Mac, PC, iPad, Brian. Oh, we might be at a, yeah. Okay, so some of you like Robert, Brian, Malia, you got, yeah, oh, ThinkPad, Alyssa, I think Anthony is probably in the best position with the desktop PC. Kevin's got a PC. Alyssa, yeah, so you guys might be okay. Everyone else, you're gonna kind of zone out for a little bit because this isn't gonna work. Fun with computers. Okay, if you click on the Insert tab at the very top, the Insert tab, you're gonna click on the arrow next to My Add-ons. Okay, and then what you're going to do is click on Manage Other Add-ons. For Macs, this whole thing doesn't exist. And then I think we want the Analysis Tool Pack. Click OK. Did I do that? Yeah. Analysis Tool Pack. Okay. And then click OK. After completing the above steps, we're going to click on Data and make sure that I have Data Analysis box, which I don't actually see. Okay, let me do this one more time. I'm going to go to Home, Insert, My Add-ons, Manage Other Add-ons, Analysis Toolbox. Let me move this out of the way. And then Analysis Toolbox, VPAC. Okay. Hit OK. I think I got it now. Okay. I think I've got it. And then if I go to data, I should see a data analysis. Okay. If you have a Mac, none of those things will work. None of those things will work. I don't know how to help you. If you don't have a Mac, you should hopefully have added in a data analysis toolbar. Okay. Types of statistics we use in biology. So we frequently use descriptive statistics. We frequently use descriptive statistics, things like means and standard deviations. So at another time, I want you to think about what do I mean when we say mean and standard deviation? What do they tell you? Okay. let's do a review. So this one everyone can do. Everyone can put the mean and the standard deviation. Okay, so we're going to again click equals, and we're going to start writing the average. And we're going to double click, and we're going to highlight and close the parentheses and hit enter. And then we're going to do the standard dev and hit standard diva and highlight and close the parentheses and hit enter. Okay, and then we're going to highlight the two, go to the bottom right corner, get the black cross, and drag and fill. So now we've gotten the mean and standard deviation for all of these things. Now, typically in a lab report, you're going to write your results as mean plus or minus the standard deviation. So you would write 15.45 plus or minus 0.7, okay, for the control group. And you should be able to do the same thing for auxin treated plants. We're going to control copy, put it here, control paste. Again, it copies the formula, okay? So here you should be able to tell me mean plus or minus, and you want to keep the units at the end. So 15.45 plus or minus 0.72 centimeters. Okay, give me 1 second, guys. I have to set up the student. See if you can get to this spot and download that extension. I'll be back in 2 minutes. Okay. You want to leave your bag here? On there, I'm going to set you up in the class. Okay. Do you need a pencil? You do. You just need a pencil. Okay, I'm back. So fun and exciting. Okay, so let's move to the next thing. 2 tips. So inferential statistics is when we want to compare 2 data sets and we want to know, are these different? And if the difference we see, is it due to randomness or are they actually different from each other? So we treat a mouse with a new drug and we compare it to a control mouse. Is the difference we see due to that drug? And for that, we usually do a student's t-test, okay? A student's t-test. So we click on the next tab here, hypo. We want to see which of these hypotheses are true. So we can either say that the differences are due to random chance only, or that they're actually different due to our experimental manipulation. The first one we call our null hypothesis, and the second one we call our alternate hypothesis. And so we want to use some data to see which of these hypotheses are true. Okay, so if you're performing an experiment, we're going to skip this step for now, but you could think about what your hypothesis would be for a plant treated either control or with auxin, which is a drug. Okay, so a student's t-test tells us if we can reject our null hypothesis. Okay, and it gives you a p-value. The p is probability, and it represents the probability that our null hypothesis is correct. So it tells you the probability that differences observed are due only to random chance. So if you get a p of 0.34, that's a 34% chance that the differences are random. They're just due to random chance. We want to set our p-value something that we think is significant. And in biology, that is typically set to a p-value of 0.05 or less. That means there's only a 5% chance or less that what you observe is due to random chance. So that's all the time when you say p-values, okay? So think about these different p-values and which ones will call us to reject our null hypothesis, which ones will call us to reject the fact that it's due to random chance, and which ones would therefore be statistically different, significantly different. Of these 3, only this one is statistically significantly different and will call us to reject our null hypothesis. Okay, so we're going to perform that. We're going to click on any field that has a number in it. And then we're going to click on our data tab at the top. And then we're going to go to our data analysis toolbar. Okay, and so this has come up, and now we've got all sorts of data analysis that we could do. They're alphabetical. We're going to go to our t-test, and we want to do 2 samples assuming equal variance. Now what this means is that the spread of numbers is the same in each group. We don't have a skewed data set, and we have 2 groups. So we have 2 samples assuming equal variance. We're going to click on OK. And now we're going to select which is our variable and which is our control. So variable 1 range, we're going to highlight these pink fields. And then we're going to click on variable 2. And we're going to highlight the blue fields. So this is control treated versus oxen treated. The alpha is what we want to set as our threshold. So we want it to be 0.05 or less. And then we're going to click OK. Bam! Look at this. Look at this math that just appeared on a new sheet. So it tells us the means, which we already calculated, the variance, and then it will go down and it will give us a p-value. It will give us p-value. Look at that p-value. Look at how small that is. So if I make this bigger here, we can see p-value on 1 tail, p-value on 2 tail. We're going to do the 2 tail. So this is telling us that we can reject our null hypothesis because they are statistically different. So if we go back a sheet, we're here, perfect. Great. So 1 tail or 2. Now on the sheet, we have our 1 tail, we have our 2 tail. When do you know which one to use? Okay, before you can determine whether they're different, you need to know if they are 1-tailed or 2-tailed. The hypothesis that you develop for your experiment will determine which is appropriate. A 1-tailed is used when you hypothesize that a treatment will cause the mean of your experimental group to be higher than the mean of your control group, or that it will be lower than the mean of your control group. For example, if your hypothesis is that you treat plants with a hormone and they will grow larger than untreated plants, you are hypothesizing that the mean of your experimental group will be higher than the mean of the control. So this means that you're going to shift the mean either above or below. That's a 1-tailed test. A 2-tailed test is that you hypothesize that the mean of the experimental group will be different, either higher or lower than the mean of the experimental group, but you have no preconceived notion of whether it will be higher or lower, only that there's going to be a difference. If you treat it with a hormone and you have no idea if it's going to be above or below, then you go with the 2-tailed test. Okay. So Excel is going to give you the results of both, and you use whichever one you've determined is appropriate. Okay. For most of this tutorial, we're going to assume a 1-tailed t-test. We're going to assume that we knew that this plant had a positive impact. This hormone had a positive impact on plant growth. Okay. We're going to the next one. Statistically different or not. So this didn't populate right, but we're going to take our values from sheet 1 and we're going to highlight them. And we're going to hit Control C. And then we're going to go back to our tail page, put it in the top here and hit Control V. Look at that. It's the same numbers. We've just copied them. Okay. So then we can look at our 1-tail number here. And we can ask ourselves, is this bigger or smaller than 0.05? That number is a lot smaller. So these are statistically significant. Okay. This is just asking you to look at them, the t-stat. We're going to again paste our values here. Compare our t-stat with our critical 1-tail. If the t-stat is greater than our 1-tail, then they are significantly different, which we know from looking at this number, right? The easier way is to look at this number here in pink, right? Which is what I said. You want to look and see if this number is less than 0.05, which means we have a difference, that there is some sort of treatment happening. Okay, and this will develop our report. You would copy and paste those numbers one last time into the report. Now, again, all of this use that data analysis toolkit that is only for PCs. Mac people out there, of whom half of you are Mac people. I got nothing for you. You know, they don't make this toolbox anymore for Macs. So I'm sorry about that. You can use a school PC. Excel itself can do a few of these things. You can do a t-test in Excel, but it's not going to give you the same format that I just went over. Okay. Any questions about that second page? Okay, we're going to do one last thing. And this, I think, is the most helpful. We're going to make some graphs. So I'm pulling up Excel tutorial 3. I'm going to, give me 1 second. Let me adjust the size so that I can see everything and see you guys and all of that good stuff. Okay, so again, we're going all the way to the left. We're clicking on that intro tab, and it's going to tell us what we're going to do. Name tab, this is where you put your name. T1, we're going to make a table, and we want our table to be a graph of the means and the standard deviations. So typically, you're going to put your independent variable and set it up so that your repeats are going in columns, okay? The next page, we're going to see a table for a figure, okay? So this is our data entry table. When you collect data, you need to have a data entry table. Most of the time, you just write this in your notebook. This is where you put all of the different replicates that you've done, right? And you calculate your means and your standard deviations. But when you make the graph, oftentimes you want to graph the mean as opposed to graphing all of these numbers, right? And so in your data entry table, the goal of it is to do your statistics. The goal of your data entry table is to do statistics. And then in table 2, we're going to make a table for a figure. Okay, so these are our raw data. We've done our statistics. We've then copied and special pasted the values to get our statistics in a table by themselves, okay? In a table by themselves. So does everyone see that? We're going to take these numbers, we're going to put them here, copy and paste the values, okay? And we now have the things that we've measured. We've measured length, we've measured where the first internode is, we have our mean and our standard deviation. Okay. We also have control and treated. Which of these things here is our independent variable versus our dependent variable? I want you guys to take a minute and think about that. What is our independent variable and our dependent variable? What are we changing? Right, so we're changing treatment. Our independent variable is always going to go in that first header here, and it's going to specify what goes in our rows. Our independent variables are here. These are the things we're changing. And we're measuring, and these go in our columns, the things that we're measuring, the dependent variable, length, and first internode. Okay, and this is the best way to set this up. So let's see if we can do what we just did. So again, we're going to click and highlight, Control C for copy. And then we're going to paste them here, and we're going to paste just the values. And then we're going to do the same thing. Control click, Control copy, and paste just the, oh, I didn't do that right. Hold on. I didn't undo. I didn't click the values page, just the values. There we go. And click the words. And we're going to write the words here. Control. And then this is not auxin treated anymore, but cytokine treated. And treated. Okay. And on the top, we're just going to copy these words here. These are what we're measuring. Control C. Oh, I shifted this wrong. I got to shift this down. Control copy. Let's shift this down. Control V. And then the words here at the top. Control copy. Control V. Okay, so now we've made our table. So this is our raw data. And from our raw data, we've made a table we're going to use to make the figure. Okay, we have multiple types of charts that you can use. Commonly, you'll see either a column chart, otherwise known as a bar chart, and an XY scatter plot, which can be a line graph. Now, these are used for different types of data. When you use a column chart, what you have on your x-axis is a description. When you use a column chart on the x-axis is a description. You see control treated, oxen treated. There should be no numbers here. You want a descriptive word. On the other hand, if you're doing a scatter plot, an XY plot, both your X and your Y axes are numbers. Words, numbers, words, numbers, descriptions, numbers, okay? And that's because a line graph implies that everything on that line exists. So there is a treatment in grams that is between 0.0019 and 0.0009. And that it is true if these are numbers. If this was a line, that would imply that there's a value in between control and oxen treated, which is not a thing, which is just not a thing. There is no value in between them, okay? So that's when you would use a column versus an XY. All great figures plot the mean. They show you the standard deviation in an error bar. They have a caption that is descriptive, which allows you to understand the purpose of the figure. You need it to be longer than 5 words. You just don't want to write oxen treated, right? Effects of oxen on total length and length of first internode in mung bean plants. Your axes have titles, and these titles also include units. Titles include units if applicable. And then you have a legend, which tells you what each of the bars represent. A wonderful, wonderful figure. If it is by yourself, it might also have a title. But typically in a lab report, it's not by itself, so it doesn't have a title on the top. But everything else is labeled. Are there any questions at this point? Any questions? Okay. Let's work on making those figures. So when you do Excel, it will put the chart kind of wherever it wants, and it typically is in a bad spot, but you can move it very easily. So you're just going to click and drag it to where you want to go. So click and drag that over to the blue box. Okay, let's see if we can make a figure. So this is our table that we've calculated the means and the standard deviations for. We're going to first highlight the column that includes the treatment names and the means only. We're not going to highlight the standard deviations. Remember, to get the second one, we're going to have to do the control. Okay, so we're going to highlight this first one, and before we let go of our mouse, we're going to hit control and then highlight the second one. On the top, we're going to hit insert, and then we're going to hit charts, and we're going to hit a column chart. We're going to do a 2D clustered column chart. Look at that. We now have total length and first internode. So you might notice that this is wrong, right? We wanted, instead of it to have the length and the first internode as our description, we want it to be control and oxen. We can do this by switching our rows and columns. So under chart tools, you should see something that says switch row and column. Click on that button. Oh my gosh, look at that. Look at that. How nice is this? So we're going to click and drag this conveniently down here. Okay. We're just going to kind of like shift it like this. So there are some pre-designed layouts for you that you can use in Excel. And they have some that are nice and some that are kind of weird. You can play around with that. For now, we're going to just kind of delete the chart title. Oh, that was not, I didn't want to delete the whole thing. Hit undo. Okay, I just want to delete the title. There we go. Because I don't think we should have a title in it. Okay, because then again, in a lab report, you're not going to have a title. And now we need to have axis titles. So you're going to click on your chart. You're going to right click, and you should be able to, oh, hold on. It's probably in chart tool. So click on your chart and then go to design, add chart element. We're going to add axis title. We're going to add a horizontal title. So this will be treatment. And then add chart element, axis titles, a vertical title, and this is going to be total, so length in centimeters or whatever the case may be. Okay, so now we have a graph of the means and it's labeled, and you can change the colors and you can kind of mess around with it, but it looks nice. But now we need to put the standard deviations in. So we're going to go to the next page. Now we have to add the standard deviations. This is the same thing, just with different colors. So the next thing we're going to do, let me just make this a little bit bigger. Wait 1 second, guys. Okay, is we're going to add error bars. So we want them to be the standard deviation. So we're going to click on our chart. Under chart tools, we're going to click on add a chart element, error bars, and more error bar options. Now, if you click on standard deviation, what it's going to do is not do the standard deviation of your whole data set, but do the standard deviation of those 2 numbers, which is not what we want, right? That's incorrect. We're going to click on more error bar options. Which one do we want to start with? The total length. We're going to hit OK. We want them to be in both directions. We want them to have a cap, and we want it to be a custom value. And we're going to specify that value as, this is where you click on your error bar numbers. Specify the value. You're going to click and highlight for positive, and you're going to click and highlight for negative, and you're going to hit OK, and you have just added in your error bars. How wonderful is that? And to just kind of double check that they're right, the control is a smaller number than the oxen treated, and that's what we see. How wonderful is that? Okay, now we're going to do it again. We're going to add a chart element. We're going to add error bars, more error bar options. Oh, we're still highlighted. Click on the chart itself. Add error bars, more error bar options. For the other one, we're going to click OK. That's fine. That's fine. None of these things. Custom. Specify the value. Highlight those numbers. Get rid of this thing. Highlight these numbers. Hit OK. Oh, forgot to get rid of it. See my thing, I have an error. Let's get rid of that. Hit OK. Bam. Now we have our error bars. We're gonna get rid of this. Okay, so this is now full of error bars that are the standard deviation. Now your figure is technically done. The last thing you could do is things to make it pretty. So in here, I have ways to change the format for the legend, the axis, the data series, right? All ways to make it a prettier graph. You can add grid lines, all sorts of things to prettify your graph. And at the end, you will end up with a figure. Okay. So that is the brief overview of how to make a graph in Excel. Are there any questions about using Excel for graphs? So again, we went over Excel basics. We went over a PC-centric view of statistics, and we went over making graphs. Because a lot of you have Macs, I'm going to expect that you can do the Excel basics and the making of a graph, and I'm not going to expect as much about the statistics, though there are some tricks on a Mac that I could show you guys next week when we're working with the Blackworms, and I have a Mac with me. Are there any questions? Okay. What I want you to do this weekend is to read about the statistics and take notes, okay? And you don't have to submit anything for this lab. So I'm not asking you to submit those final pages. This is mainly because when we finish the Blackworm lab, you're going to have to do a lab report, and you're going to have to make graphs for your lab report. And so I want you to be able to make pretty graphs, okay? Any final comments? Okay, you guys are good to go. As soon as this video is set, I will post it for everyone. Otherwise, I will see you guys, Group B, I will see you tomorrow. So last names towards the end of the alphabet, tomorrow virtually on Zoom. I will send out that link this afternoon. Otherwise, Group A, I will see you on Monday. You guys are good to go.
Professor Carroll, you said that we didn't have to send you like the screenshot